Yeah. She is Dr. Sandra Catherine Janithan. She is currently working as a lecturer and researcher at the Faculty of Fisheries and Food Sciences, University of Malaysia, Terengganu, Malaysia. She is also the program head for Bachelor in Aquaculture Science in her faculty. She received his doctoral degree in aquatic virology from University of Tasmania, Australia. Dr. Sandra has authored several publications in various journals and books. Her publications reflect her research interest in viral diseases in diversity of aquatic organisms, including finfish, oysters, mussels, shrimps, and coral ecosystem. Her topic of presentation is molecular epidemiology of Megalocytivirus in Malaysian freshwater angelfish, Pterophyllum scalare. Now I request to play the presentation of Dr. Sandra.
Good afternoon to all. I'm here today to present one of my master students' project entitled Molecular Epidemiology of Megalocytivirus in Freshwater Angelfish Pterophyllum scalare from Malaysia. This work belongs to Cik Azharul Zaman. The ornamental fish industry in many Asian countries are developing due to its high demand, either at the domestic or international markets. Malaysia is one of the world's top producers of ornamental fish, where the products are being exported to China, Singapore, United States, Japan, and Iran. This also contributes to the Malaysia's export value. In 2020, the Malaysian ornamental fish industry production was at 227 million tons, valued at almost 500 million ringgit Malaysia. There are more than 250 species of 550 varieties of ornamental fish being cultured in Malaysia. Frequently exported fish include smallies, guppies, bettas, arowanas, and so on. These ornamental fish are affected by a viral disease known as the Megalocytivirus infection. The sources of this infection and the transmission are unknown in Malaysia, since the ornamental fish industry is a million-dollar industry. Thereby, it was vital to tackle this issue. Megalocytivirus infection in ornamental fish in Malaysia causes non-specific clinical signs. Thereby, it was difficult for the farmers to observe any clinical signs at the farm level. When these products are being exported to overseas location, they are being tested as Megalocytivirus positive at the quarantine area. So in the future, the export of ornamental fish from Malaysia may be reduced when if there was frequent occurrences of Megalocytivirus. There have been reports of high level of mortality associated with the presence of Megalocytivirus in many different freshwater ornamental fish. Thereby, the objectives of this study were to detect the presence or absence of Megalocytivirus in one of the popular ornamental fish, freshwater angelfish, to determine the genotypes of Megalocytivirus, and lastly to determine the major risk factors that contributes to this disease. This is the taxonomy of freshwater angelfish. It belongs to the Cichlidae family. This is the taxonomy of Megalocytivirus. There are many genotypes of Megalocytivirus. One of the best known Megalocytivirus in ornamental fish are ISK-NV and RSIV. Megalocytivirus is an envelope virus and the diameter is between 120 to 300 nanometer. In our previous findings, we have tested Megalocytivirus positive samples in 10 to 15 species of ornamental fish. We've also found there were also reports of positive Megalocytivirus in food fish such as grouper, mussels, oysters, and also trash fish used for grouper in Malaysia, and they belong to ISK-NV. This study was done at an ornamental fish farm in Johor, and this is the plan of the farm where the study was done at the broodstock pond, breeding site, as well as the grow-out pond. 7 pairs of broodstock were used for this study. So we also used the gill and mucus tub as swabs for the broodstock, and a study was done, followed the routine farm production from broodstock up to the market size of the fish, day 45 or day 60. We also conducted some preliminary sampling because we wanted to look at the detection of Megalocytivirus in environmental samples such as water quality parameter, water samples up to snail and snail eggs. So this is the non-invasive sampling method which was conducted for the broodstock. So we used the mucus and as well as the gill swab. The sampling days followed day 0 up to day 60. A total of 935 samples were collected, ranging from fish samples to environmental samples. DNA extraction was conducted according to GF1 extraction kit. We used the Riemer and Whittington nested PCR analysis for this study. All the samples were processed accordingly. Then once DNA bands were obtained at the gel electrophoresis stage, these samples were proceeded and sent for sequencing analysis. Once the sequences were obtained, phylogenetic analysis were conducted. Pearson correlation coefficient were used to determine the strength of correlation between the presence of Megalocytivirus in freshwater angelfish with fish age and also other environmental samples. So in this study, we also looked at the biosecurity and hatchery practices which were implemented by the farm, and some of these practices ranging these aspects were not implemented in the farm. So according to previous reports, some level of biosecurity practices is important to prevent the Megalocytivirus infection. All the water quality parameters were normal except temperature at broodstock pond and nitrate, nitrite and ammonia level. So in the previous study, similar level of water parameters have been known to cause positive Megalocytivirus infection in ornamental fish. There were no clinical signs observed in all of the fish, so all of the fish looked healthy. This is similar to previous study where Megalocytivirus is known to cause non-specific clinical signs such as lethargy, skin lesion, loss of appetite, ulceration, and etc. Okay. A total of 312 samples were positive for Megalocytivirus, ranging from the water samples, the swab samples, mucus and gill, the fish samples from eggs to day 60, sediment, live feed samples, and wild fish. Only snail ate, and one of the wild fish, which is Psilia reticula, did not produce any positive samples. These results also indicated early detection of Megalocytivirus infection at the egg level. Positive water sample also indicated some evidence of horizontal transmission and also other possible viral transmission routes from other environmental samples. Once the farmer had changed the live feed supplier or source, then there were no more presence of Megalocytivirus in the farm. So other studies have also used a nested PCR assay. There were no clinical signs and no positive band at the primary reaction of the PCR analysis, and this is due to the asymptomatic carrier of these affected fish in Malaysia. It does not cause symptoms thereby, but they are able to transmit and infect other fishes. This is also the reason why we use the nested PCR analysis to detect the carrier status. The sequencing and phylogenetic analysis showed that all of the nucleotide sequences in the study showed highest similarity to ISCAN-V from 98 to 99%. So this is also similar to previous reports found in Malaysia for Megalocytivirus. Most of the freshwater ornamental fish which were positive, they belong to the ISCAN-V. So according to our epidemiological data analysis, mostly we found moderate and positive relationship showed between the presence of Megalocytivirus with high temperature, concentration of nitrate in water sample and high DO and pH in the fish. We also found some negative relationship between the virus with ammonia, level of ammonia, level of nitrate and nitrate. And there was some negative association of Megalocytivirus when the DO and pH of water sample and temperature and DO for sediment samples were low. But all of the risk factors showed significant statistically.